first of all, welcome everybody to this month's um, California Botanical Society seminar. I'll get to that in a minute. In the meantime, I just want to remind everybody of the upcoming Graduate Student Symposium, which will be at Cal Poly Cal Poly San Luis Obispo. Kathleen, when is it?
It's Saturday, April 5th for the Grad Student Symposium with the banquet that night, but we will have field trips on Friday and Sunday, kind of bookending the symposium. It should be an awesome weekend.
Yeah, it's going to be really great. The organizers have really thrown themselves into it, and it's going to be a fantastic event. So,
And
there
your students.
and of course, and your students, and your students. Um, then let me go ahead and announce today's speaker, Allie Richards. So Allie has actually, she's a graduate student right now, a PhD student at UCR, but she already has an extremely lengthy CV. So I'll just try to sort of summarize a few of the things. She attended Saddleback College and then got a BS in biology with an emphasis in botany from Cal Poly Pomona, working with Erin Questad. And then since 2021, she's been a PhD student in plant biology with an ecology focus at UC Riverside, working under Dr. Ezekiel Escura. While she has been doing that at UCR, she also has obtained a certificate in curriculum design, a university teaching certificate, an artistic expression of original research certificate related to SciComm at UCR, and a science to policy graduate certificate. She's currently serving as a STEM consultant to the UCR Teaching Assistant Development Program and graduate student representative to the International Affairs Committee of the Botanical Society America, as well as graduate student member of the UCR National Reserve System Advisory Committee. But wait, there's more. At the same time, she's a graduate student intern, including giving a guest lecture in plant diversity and ecology at Santiago Canyon College through the California Community College internship program out of UC Irvine. In her spare time, she's chair of the UCR Science to Policy Initiative. She organized a biostatistics in R workshop. She's been president of the Botany Graduate Student Association and an organizer of the annual Botany and Plant Sciences Symposium at UCR, as well as executive board member of the Society for Ecological Restoration, the UCR chapter. She's got an equal number of many awards and fellowships, including Outstanding Performing Artist Fellowship, Outstanding Teaching Assistant in Evolution, Ecology, and Organismal Biology at UCR, and she was the winner of the 31st Botany and Plant Sciences Graduate Student Achievement Award at UCR. That's just maybe a third of what's on her CV. So I asked her what she likes to do outside of research, although you can actually, you've already heard about some of it. I know that one thing she's really passionate about is teaching and mentoring, really passionate. And when I asked her what else she's passionate about, she responded that she's the chair. She's passionate about chairing the Science to Policy program at UCR. She also is passionate about cooking, indoor cycling, macro photography, which you'll get to see in her seminar, interior decorating, and cafe hopping. Particularly, she says, I love hunting for a good iced salted caramel latte. So now you know Allie, and with no further ado, let me turn this over and she can begin her presentation on extrafloral nectaries in Ferocactus. Take it away, Allie.
Thank you so much, Amy. I don't quite realize how long my CV is until someone else reads it out. Let me go ahead and share my screen here. Let's see. We'll get started. Okay, and can everyone see my screen all right? I'm gonna go with
Yes, I can see
okay.
it.
Okay, Awesome. Wonderful. Well, thank you all for having me. I really appreciate being invited to speak to you today. And just like Amy said, my name's Allie. I'm a PhD candidate in the Escudo lab over at UC Riverside. And today I'll be talking to you about the sweet secrets of cacti, how Ferocactus uses nectar to survive and thrive. So today's flow, got a couple different sections here for you today. What are extrafloral nectaries? Defensive mutualism, extrafloral nectaries in cacti, Ferocactus, the North American barrel cacti, and my research in Ferocactus. So you may be wondering, what are extrafloral nectaries? Before we cover that, we really want to learn about the traditional nectary that is discussed, the intrafloral nectary, and its role in reproductive morphology. So it's typically found at the base of the flower here. And it's used in to aid in pollination. So it coerces pollinators deep into flowers. So the pollen can get stuck to sticky stigmas on their way in and out of flowers. Now, that's not necessarily the role, though it can help with pollination. It's not the role of extrafloral nectaries, and they can be found in many different forms, including sunflower phyllaries and the stems of the acacia bullhorn. And only some plants have extrafloral nectaries, not all of them, but extrafloral nectaries, or EFNs for short, are nectar-secreting glands that are located outside of flowers, often on stems or leaves. Now, they aid in a type of symbiosis, and I want to cover that word as well so that we can really get on the same page here. But symbiosis is basically an interaction between two species, and there's three main types of symbiosis. And the first is parasitism, where one species benefits and the other is harmed. And we're all familiar with parasitism, especially. I don't know if you're all from the Riverside area nearby, but this summer, mosquitoes get really bad over here, and that's one form of parasitism. The next type of symbiosis is commensalism, where one species benefits where the other is unaffected. And my favorite example of this is in whales and barnacles, where the barnacles are benefiting through filter feeding as whales pass through the ocean, and the whales are completely unaffected. And then my absolute favorite type of mutualism, or type of symbiosis, is mutualism, where both species benefit. And this is the one that's seen between extrafloral nectaries and ants. So when we have this relationship where some plants have extrafloral nectaries, they're secreting nectar outside of those glands. They're not looking for pollinators. They're looking for bodyguards. They're looking for defense. So these bodyguards participate in an exchange of goods and services, where the plant provides food and shelter while the ants provide a defense against herbivores for the plant. And this is referred to as defensive mutualism. So you have defense against herbivores in exchange for that food and shelter. Now, this is just an example on the Quazon cherry, where ants are actively seeking out nectar just below the leaf. Another really popular example of defensive mutualism in the ant-plant relationship, and one that has absolutely saturated literature, is the relationship between the acacia bullhorn and acacia ants, named exactly for this relationship, where ants actually hollow out the thorns of the acacia bullhorn and use it for shelter, while eating at the Beltian bodies that are found on the stems of the plant to keep away herbivorous insects that threaten, eating any sort of leaves that are on the plant, fruits, and just overall affecting the health of the plant if the ants weren't there to protect it. One other thing that they do is keep those other invasive plants off of the host plant. So some acacia bullhorns that may be growing nearby and invading the space of the current host plant, ants will actually eat away at the smaller plant to keep it from competing for resources. So protection on all sides. So one thing I kind of touched at the very beginning was the interaction that ants can have in pollination. And so those plants that don't have extrafloral nectaries, there's been literature that shows that they actually start to impede on pollinators' ability to effectively do their jobs. At that rate, they're nectar robbers. And so ants or plants that do have extrafloral nectaries actually act as a that nectary acts as a distraction to keep ants away from intrafloral nectaries and allow pollinators the chance to really do their job effectively without interruption. And one of my favorite studies that shows this is the one from Villamil in 2019. Where they did a control versus clogged experiment of floral and extrafloral nectaries on a plant during the pollination season, and it was noted that the ants near the EFNs it significantly decreased while they went right towards the intrafloral nectaries in the clogged treatment group. So when it comes to the presence of extrafloral nectaries on cacti, we have to first look at the evolution of spines. And spines didn't always exist in nature the way we see them now. There's a few generations removed leaf, actually. So leaves over time started to shed away and become spines, and we see this in a recent ancestor, a common ancestor with leaf plants in Pereschia, and those leaves eventually fell in other species so that we only have spines on the stem. So these are simply generations removed leaves, if you will. And those spines are a first line of defense against larger herbivores, typically. And I say they mostly protect against larger herbivores because they don't always do a great job, especially with those bighorn sheep in a drought year. Those horns are willing to do just about anything to get to the center of a cactus. But then when those spines don't defend against insects, herbivorous insects that eat away at flowers and fruits, we need a second line of defense. And those spines that are no deterrent for these smaller herbivores, extrafloral nectaries have then become this evolved second line over millions of years in response to the herbivory. So the purpose of the extrafloral nectary is to attract territorial ants that then scare off or fight those insects to the death. And it's like having bodyguards that work for sugar. So now we want to talk about the evolutionary elongation of the areole. And this kind of comes in tandem with the evolution of spines from leaves. So once we have these stems that are entirely spines and areoles rather than any leaves present with their axillary buds, those axillary buds are actually, the areoles are also transformed axillary buds. And over time, the areole has expanded to include floral meristem as well through that evolutionary history. And it's further expanded in some species like Ferocactus, some genus, some genera like Ferocactus, to also include space for extrafloral nectaries. And with this elongation, we've been able to tie those extrafloral nectaries to spines. So in the evolutionary development of extrafloral nectaries, you have leaves, spines, and then our nectaries on those areoles. And in cacti generally in the family, there's only about it represents only about 3.5% of all cactus species that have extrafloral nectaries. So we only have about 156 cactus species that show extrafloral nectaries somewhere on the plant. And there's 4 main types that have been identified. The first of which are extrafloral nectaries that are modified spines. They're short, broad, and blunt. And then the second are nectaries that resemble spines, but those nectaries secrete just along the spine. And then we have small foliage leaves that occur below the areole along floral tissue. And then regions of epidermis below the areole as well. Now, something you may notice about 3 out of the 4 of these types is that they're only apparent because we can see the nectar droplets forming. If you really take a look at that first image with the extrafloral nectaries that are short, broad, and blunt, there is no nectar present. So we can only see them because they kind of stick out. And for that reason, it makes Ferocactus an ideal species, a model species for this work. So this is the one that I'm most interested in. Now, like I said before, these extrafloral nectaries are the ones that are observed in Ferocactus, and Ferocactus appears in many different forms. They come in sizes ranging from 2 to 10 feet tall. They grow in rocky, well-drained soils on slopes. They're generally globose. They're barrel-like or cylindrically shaped. And their fruits and flowers are vibrantly colored, while their spines can be long and prominent or even fishhook shaped, which you'll see in just a second here as well. And those spines can be red, yellow, brown, shades of gray even. We've got some of my favorites here, those being Ferocactus robustus, Ferocactus hystrix, Ferocactus latispinus, Ferocactus viridescens, and Digwettii. All of these range throughout the American Southwest and into Mexico, which we can see here in, this was a quick range map that I made in just the last year. So you can really see just the diversity of species within the genus found across the Southwest and down into Mexico. And these species are primarily found across 3 North American deserts that include the Sonoran, the Mojave, and as well as parts of Baja California and down into central Mexico. Now, this includes all of the current distributions for Ferocactus, but I did leave just about 3 species off based on their status with the IUCN Red List as endangered. And I'm most interested in the species that grows in the Coachella Valley and parts of Joshua Tree. And that one is the California barrel cactus. And that guy was just at the top there, sorry. There are a few really important things to note about Ferocactus in general, and one of those is the ethnobotanical uses that this genus has. Many of the species are consumed raw or cooked. In some species, there's some claims that they improve digestion, brain function, or even blood sugar, as well as the physical attributes of the cactus that you can use them hollowed out and as a cooking pot. You throw some hot stones in there. And you can use the spines for tattooing, sewing needles, and fish hooks. Many indigenous groups in the southwestern United States, as well as throughout Mexico, have used these species for those reasons. Now, there's a lot of broader implications when it comes to the work that I do in Ferocactus. And the first of which is ant plant mutualisms. They maintain populations of seed dispersers. So those ants that are now finding a new food source with extrafloral nectar, it's keeping those ants that also move seeds in the area. This work can also to help inform habitat restoration, like reintroduction, as well as conservation. So understanding if an ant species moves out of an area, does that affect the cactus population? Is that the reason some of these Ferocactus species are declining? They also increase resilience and increase nutrient cycling, especially as we have those larger mammals eating away at plants. Now, Ferocactus has been studied once before in terms of its reproductive cycle, mainly in May and June, and this was back in 1986. Ruffner and Clark looked at the buds, flowers, and fruits and when they were appearing in that reproductive season. And it was noted that the buds were very, very frequent both in May and in June. So that's showing that the plant is really trying to increase its chances of getting to that fruiting stage by producing as many buds as possible, hoping that there's enough pollinators in the area to then come and mature those ovaries. In May, we also start to see those flowers with an increase in June, but June is when we really start to see the fruits of that labor. Keeping this timeline in mind of all of our buds are coming in May, they're all starting to come in May, they're all still very heavily present in June, and then those fruits are only present in June there. We look at the developmental stages, and this was something that was done by Bentley in 1977, I believe, as a part of her dissertation, which I always thought was really cool. But if we look at this graph here that shows the ants and the rate of extrafloral nectar secretion, you'll see that the ants marked by a dashed line with solid dots there. They appear most often at mature buds, which further emphasizes the role of ants acting as bodyguards, trying to make sure as many buds as possible make it to that flower stage and eventually the fruit. Once we do get to that flowering stage, there is a decrease in nectar production and thus a decrease in ant presence. And with that, it's at least my hypothesis is not only do the extrafloral nectaries act as a mode of distraction, but they can also still look elsewhere as the nectar robbers. So keeping them entertained long enough to keep them out of pollinators' way, while also not giving them any room to go and nectar rob in a way. So giving them a sweet treat to stay out of the way, but reducing the nectar in hopes of reducing the number of ants. And then once we get back to the young fruit stage, that's when that nectar production increases once again. So we're at another vulnerable point in the plant's reproductive cycle where we need that extra layer of protection from ants. And so nectar increases and ant presence increases as well. Now, extrafloral nectaries, from a microscopic view here, this one is really interesting. This is a scanning electron micrograph of one of our EFNs in Ferocactus, and this is in Ferocactus amorii. It's broken up at the top, and it's not totally sure in this article. It wasn't totally sure if that's from ants chewing down at the top of the nectary or if it's just wear and tear over time. But you can really see the layers of epidermis inside of that nectary. It's almost like fiber-like cells. But there is one intact in the background there, and that one is, you can see very clearly the kind of almost a hole in the top center there, and I believe that's where the ants are kind of sucking up the nectar out like a straw. And the nectar that they're receiving, there's 3 different types of sugars that they're receiving from that extrafloral nectar. And those are fructose, glucose, and sucrose. And fructose, these are some of the reasons that ants go for them. Fructose metabolizes slowly and it provides a sustained source of energy, while glucose metabolizes quickly and provides a burst of energy And then sucrose is a nice combination of the two. So you'll notice I put in a little box here at the top under May. So thinking back to that graph of, what's going on in May, we have a very large amount of floral buds that need protecting, and the start of flowers that need protecting. And so sucrose really acts as like the gold that an ant can come across and find as a colony, to really drive them in and try to get them to move into the system, to protect the plant long term, so that when we move into that reproductive period in June, sucrose is decreased and we have an increase largely in glucose, to give bursts of energy, quick sources of energy, so that those fruits are still protected throughout the reproductive cycle. Now, this is one example of one of the larger examples, I should say, of extrafloral nectaries in Ferocactus. And I think this one's a really interesting example because I don't, especially in my California barrel cactus, I don't see nearly as many extrafloral nectaries per aerial. And this one, it actually has I counted around on this image here, and the one that has the most has 17 extrafloral nectaries, where we see other species range from 0 per aerial, maybe 1 on a plant, to 17 here. So with that California barrel cactus, I do see maybe 1 to 5 extrafloral nectaries, and you'll get a chance to see those up close here. But Ferocactus cylindraceus, the California barrel cactus, that's the one that's located in the Coachella Valley and Joshua Tree as well. It has bright yellow fruits and flowers, and it grows up to 10 feet, although I've never seen one 10 feet tall. I've only ever seen one 10 feet long, falling down after some time. And those spines range from yellow to red. These two images were actually both taken at Boyd Deep Canyon in Palm Desert, that's in the Coachella Valley, yet still related. So the California barrel cactus, like I said before, is the one that's found just up at the top here. This is like the northernmost part of the Ferocactus range, up in Joshua Tree National Park and the Coachella Valley. And these are the populations I'm most interested in, basically based on location, but it's also an extremely stable population in comparison to others. The 3 species that we have in the southwestern United States are relatively stable compared to the southernmost populations in Mexico. So the California barrel cactus, this guy here, it has active extrafloral nectaries that are located between floral buds and spines, and only around the crown of the cactus, so that those top few nodes that we find where new growth occurs near the apical meristem. And they're definitely more than just decoration. They're easy to spot, even without active extrafloral nectar secretions or ant presence. And that makes this a great model genus, especially this particular species with its locality, for extrafloral nectary studies in Cactaceae. I was really fortunate to grab this image, actually. This is like finding a needle in a haystack, being in the field, because if there is nectar on these nectaries, the ants are usually there first. They're usually suckling on the nectary way before I can get to it. So this was kind of a miracle of an image that I love to show off here. And the defensive mutualism in the California barrel cactus, we see 3 main genera of ants that participate in this. And the one you see on your screen now is Chromatogaster. It's one of our Chromatogaster species. But the other 2 are Solenopsis as well as Myrmecocystis, and they participate as mutualists. They love the sweet nectar and in return, they protect the cactus from cactus bugs and other insects. And that helps to increase the reproductive success of cacti in a very, very harsh environment like the desert. So this is what we like to consider a win-win. I do have a quick clip here as well. It's from the field. I do hope the volume doesn't turn on. It's quite windy in the video, but bit of a blink and you miss it. Ants are patrolling their food source on each aerial of the cactus, waiting for an herbivorous insect to come in and attack. And one interesting fact I like to throw out there too is the ants don't necessarily know that they're protecting an entire plant for the plant's benefit. Their understanding of the situation is they're protecting a food source. So it's really important to keep in mind with this type of research as well, because you'll have some that are better than others at this job. But a few of the big questions that I'm hoping to answer in my dissertation research, and I've begun to answer already, are there life phases that are attributed to extrafloral nectary development? Which ant species colonize Ferocactus extrafloral nectaries? As well as, is there a phylogenetic or geographic correlation for the presence of extrafloral nectaries within Ferocactus? Sorry, talking a mile a minute. So the first question I want to cover is that first one. Are there life phases attributed to extrafloral nectary development? And I've identified three key developmental stages when exploring the development of extrafloral nectaries. And all of this stems from looking at the stems, looking at the apical meristem at where it all begins. So this stage I'm calling formation. Happens at young areoles near the apical meristem. So we see an example here of an areole that only has spines on it. It's yet to develop any floral meristem. There are no obvious extrafloral nectaries that are located within that areole. But just one node back, we have the full trio. We have our spines, our EFN, as well as floral meristem and a forming bud as well. In the second stage, we have activation and secretion. And this stage is something that is yet to be understood in terms of triggers, whether it be environmental, seasonal, or ecological interactions that trigger the secretion of nectar. But this is the mature stage of the nectaries. And a final stage in expiration or senescence. And this is something we see just below the apical meristem. This is only a few nodes back from there. But we can clearly see scarring from previous reproductive cycles where floral tissue used to exist, where fruits used to appear. And it all kind of works its way downwards. So as the cactus is growing upwards, kind of moving in that pattern there. And that's expiration. Now, one of the things I want to understand is discerning the age that EFNs develop alongside initial floral development, while also understanding the relationship between height and cactus age. So one of the methods that I'm employing is the radiocarbon dating of spines. So analyzing the carbon-14 in spines to understand how old a particular areole may be. So I'm collecting spines at the oldest expired extrafloral nectary that I can find on the cactus. They're usually a lot lower than I'm showing on the image here, but I didn't want to bunch too many things together. So the oldest expired extrafloral nectary, the oldest floral scar, and at the base of the cactus. So I'm collecting spines at each of the areoles where these features are found. And I'm hoping to apply that to a similar age-height relationship graph to this one, which was created in 1997 by Bowers, looking at Ferocactus wislizeni, which is one found over in Tucson. looking at the estimated age-height relationship with high and low estimates, with the median in that dark black line in the middle there. But I'm hoping to reaffirm these estimations as well as identify structural development points in this age-height relationship. So maybe floral development, from what we've seen in the literature, it's about an age of 30 years that begins. But do extrafloral nectaries develop in tandem at that point, or is it just a little bit later in the age of a cactus in their life cycle? The second question that I hope to answer is which ant species colonize Ferocactus extrafloral nectaries? And I've begun collecting plenty of ant specimens to get identified over at UC Riverside. And some of them are on the screen here, being Solenopsis geminata, Chromatogaster, and then we have 3 different species of honeypot ants. Now, these honeypot ants have yet to find an expert that can really get those down to the species level, but we're hoping to get those identified visually, as well as potentially do this through DNA barcoding, especially for those honeypot ants that have yet to be identified. But I believe I'm looking at 3 to 5 different species of honeypot ants that are participating as mutualists. And these are the same ants that I will continue to assess. I want to understand their defense, their level of defense in this interaction. I'll be doing that via manipulation, trying to see how effective of a mutualist they are. Something about ants is there's a dominance hierarchy within them. And so the more dominant, more territorial ants will usually win out a territory, including a cactus territory, and will be a more successful defender of that plant because of their aggression. Where others that, may have found a cactus that hasn't been occupied by a more dominant ant species may not defend as effectively as others. So in addition to assessing their effectiveness, I also want to see how they contribute to the soil, both in the nutrient contributions as well as their soil aeration contributions. So this I'm hoping to also overlay when it comes to those ranges of ant species, their distribution ranges, overlay some of their distribution ranges with the Ferocactus ranges to see how they may be interacting not only in the southwestern part of the United States, but maybe these same species are found in areas where there are decreasing populations of Ferocactus, or they used to be apparent in those areas, and we can potentially tie together those two to assess why a population might be in decline. Now, one of the other attributes I discussed was assessing the nutrients that the ants may be contributing to the soil. And so I've collected soil samples from my different cactus species over in Boyd Deep Canyon at 80, 30, and 0 centimeters away, so at the base of the cactus, to run isotopic ratio analysis on. So just understanding the isotopic versions of nitrogen and carbon and how they may be present in the soil, and if there's a gradient when it comes to that. So the hypothesis behind this particular study is that ants, while they nest at the base or near the base of cacti, may be contributing excess carbon and nitrogen to the soil that could benefit the cactus. So it's like an added benefit on top of the protection that those ants are offering. And another one that I want to focus on too is that soil aeration factor. And this piece is actually a sculpture done by Walter Schinkel. I think I nailed his last name this time, actually, saying it again. But this was one ant nest. It's a aluminum cast of Solenopsis invicta, and this is a cousin of one of the ant species that I've found on my Ferocactus. And this nest is underground, just below plants. It's over 6 feet, in length, and it's extremely dense in terms of the different rooms that they allocate the resources in, which is part of the reason I've hypothesized about the nitrogen and the carbon is they have waste rooms or bathrooms, if you will, as well as like food storage rooms. And these rooms are forgotten about over time, and the soil reclaims the nutrients that are brought into these ant nests, into these colonies. So with how dense the colony is, I hypothesize that there's an increased soil aeration, and thus an increase in water infiltration. So this is critical in the desert because rainfall is few and far between, especially in those summer months. And so the deeper that rainfall can penetrate, the better it is for the plants in the desert, especially our cacti. So if they're increasing soil aeration through these very dense colonies underground, cactus roots may be more easily able to obtain rainfall as a mode of capture. So one of the ways that I'm testing this out is through water infiltration rate experiments. And this one hasn't quite started yet. I'm hoping to get this done soon. But we'll be categorizing the soil type so that I can locate the basic infiltration rate that needs to be compared to when we do these measurements. And it's relatively straightforward in terms of ranging from sand to loam to clay and in between as well. And these experiments are typically done with a double ring infiltrometer, although we'll need to construct something a little bit different, modified, so that we can get closer to the base of the cactus and work around those cactus spines to get the most accurate measurements possible. So this is just one of a few things that I think is going on in addition to that protection of the cactus. Now, in my last question, I want to ask, is there a phylogenetic or a geographic correlation for the presence of extrafloral nectaries within Ferocactus? And so one of the things I'm looking at is an already constructed phylogenetic tree that we have from 1997. This is the most recent tree that we have that just shows off Ferocactus, and it only has about 15 species, so we're missing roughly 10 to 12 species, I believe is my last count. So quite a few are missing here. I don't think I'd get the chance to do it, but reconstructing it would be quite the challenge, the mission, so that I can then compare and see how many extrafloral nectaries are found throughout the phylogenetic tree and what that pattern might look like. So thinking back to our Ferocactus peninsulae, where we had those 17 extrafloral nectaries, is this a more recent member of the phylogenetic tree, or is this more of a historic member of that phylogenetic tree? So trying to find an association of the presence of extrafloral nectaries with their positioning in that tree, maybe seeing an increase or a decrease in the presence. I also want to look into the geographic correlation of these. So same map again, but wondering if the average extrafloral nectary count per aerial increases or decreases with the ranges from south central Mexico all the way up to our southwestern United States. Will we see an increasing presence of extrafloral nectaries based on those locations? So I'm planning to overlay the data on that map as well. I'm going to end there and say, in summary, extrafloral nectaries help Ferocactus to survive and thrive by recruiting tiny ant allies, but we still have a lot to learn about these very important structures. And I want to give a quick shout out as well to my lab members, one of which just recently graduated, as well as my funding sources, some of which Amy had mentioned before as well. And give a quick thank you with one of my favorite macrophotography images that I've taken. But love to take any questions you have.
Well, thank you, Allie. That was really fantastic. I love hearing about your work, and extrafloral nectaries are just such an unexpected thing. And to be honest, I never actually imagined them on cacti. So
Right.
this is really informative. If you
Thank
have
you.
questions, you can raise your hand, as I see Peter has already done. Feel free to put questions in the chat, or if you want, if there's a lull, you can just unmute yourself. So, Peter, what is your question?
Hi, thanks so much for the talk, Allie. Does floral nectar differ from extrafloral nectar?
That's a very good question. Not by much from the studies that I've seen. They've basically got the same 3 sugars in each, although some nectars can also have proteins or amino acids as additions that may give pollinators more benefits. But in the studies that have been done in cactus extrafloral nectar, it's mainly just those 3 sugars that are present in larger quantities.
Cool. I wonder, do you know how much how many different species has the nectar from extrafloral nectaries been looked at?
Not very many. Like I mentioned before, there's only about 156 known species, and when people are looking into extrafloral nectaries, they're typically looking to those tropical plants like acacia bullhorn. They don't look to our desert species
Or yeah,
because they only have about 3.5% of all extrafloral nectary bearing plants.
wow,
So if there are studies done, it's typically Ferocactus, and there's only been a few.
yeah, cool. Michael.
So thank you. That was really interesting. At one point, you mentioned how the ants are kind of patrolling, and there might be a herbivorous insect. That. And I'm just wondering, how frequently do you see that happen? Like an unwary insect pops in and they grab it.
Um, Not super frequently, but they do. Sometimes I bump the cactus the wrong way, and I am an assumed intruder, and I really get to see how it all plays out. It's interesting though too, because they ants are a lot smarter than we give them credit for, I think, and they do recognize that I'm like a larger being, and I've seen them run down the side of a cactus and try to come up my leg rather than going after the hand that was like on top of their territory. But I've seen it only a few times. It's usually a pretty quick interaction. I would have to sit there for quite a while to see if it goes on.
Hugh.
I think he's muted.
You have to unmute yourself. Unmute yourself, Hugh. Maybe I can do it. Hold on a second. Whoops, shoot. I can't unmute you. I can only ask you to unmute.
You can try holding down the space bar.
There you go. Okay.
I
You should
don't
be
know
able to.
why I can never find the unmute setting when I want it. I see it all the time when I don't want to unmute myself. And then the moment I go to find it, I can't find it. Sorry. I just wanted to say I find this immensely fascinating, although
Thank
I know
you.
very little about ants. I've read a lot of research on spiders and on aphids. So I
Mhm.
realize this may be outside the scope of this meeting, but I'm just dying of curiosity. Has any of your research or observations included any relationship between aphids and exofloral nectaries?
I'm glad you asked this. I get this question a lot too, and I haven't seen any aphids present on my Ferocactus in general, but definitely not when the ants are there.
Ah,
Yeah, I haven't seen, because I do know that they have a relationship as well, ants and aphids, that's commonly studied. But yeah, I haven't seen it play out on cacti. And I mean, part of that might be because of the extrafloral nectaries, where ants don't need the nectar they would source from the aphids. But who knows? Yeah, it's a bit out of my realm. I just became a pseudo entomologist because of this study. I'm a plant biologist at heart.
Ah,
So yeah.
okay. Thanks.
Other questions? Thanks, Hugh. Other questions?
There's a question in the chat.
Oh, there is?
I
That's
could,
interesting. Oh, there we go.
I could ask it. It's what is written in the chat there. So
Okay,
I could.
yeah, go ahead and ask it.
So unfortunately, near the coast in San Diego, we have a lot of Argentine ants,
Yes.
which displace the harvester ants. And I'm
Yes.
wondering whether the cactus realize and stop putting effort into the extrafloral nectaries, or is there any response to lack of ants?
This is actually where my entire dissertation project began, was watching Argentine ants on barrel cacti at the UCR Botanic Gardens, because we have just invasive species over here. And the cacti are definitely still producing extrafloral nectar, and those ants are not acting as mutualists at all. One of the first hypotheses I was considering was looking at some native species of ants maybe not participating in the mutualism at all, but it turns out it's the non-native invasive species that don't participate in that mutualism. I got to watch 2 different types of flying insects eating away at flower buds while those invasive species of ants were eating away at the nectar. It was definitely sad to see. And one of those things that, you know, after looking into it a little bit further, made me think, well, is this why some of the Ferocactus populations that are vulnerable, threatened, endangered, is this why their populations are declining? Is it those invasive ants pushing out the beneficial mutualist ants?
So many reasons to hate Argentine ants.
Right. Yeah, just add to the list there.
Hugh, do you have another question, or your hand is still up?
I think his hand might be left over.
I'm sorry, I didn't realize I had to put my hand down. I'll do that.
Yep, sorry. Any other questions in the
I
chat?
have one more quick one.
Yeah.
Just I wonder if over different years, when there's been dry years versus wet years, if you see any difference in the production of the nectar.
That is something that I won't have the opportunity to look at, and I don't think anyone has just yet. But it is one of those factors to consider when it comes to that activation and secretion stage, when environmental factor like precipitation in a particular season, will that lessen the amount of nectar secreted, or it might even have the opposite effect, increasing the nectar that is secreted to increase the amount of bodyguards that they have in a drought year, because you have animals like bighorn sheep that turn to cacti in those years. So yeah, it could definitely go either way. It's not something I'll have a chance to look at, but a very interesting question nonetheless.
That can be your next project after you graduate.
Right, yeah. Let me just add to my dissertation a little bit more.
Any other questions?
Sorry, I jumped in a little late, so I didn't get the beginning, and maybe you addressed this, but I was wondering, do the ants, when they're defending the cactus, are they just like using their mandibles, or they're also stinging, or a combination?
From the few times I've seen it happen, it's just been with their mandibles, although I'm sure it varies species to species. I am just learning, I'm barely learning ants for my dissertation, and just trying to understand their social habits as best I can. But yeah, from what I've seen, it's just been their mandibles, and sometimes it's a fight to the death scenario, and other times they just scare off the herbivorous insect. So
Thank you. It was a great talk and such wonderful photos.
Thank you so much. I appreciate that. Yeah, this was mentioned briefly in the intro, but I learned macro photography for this dissertation because there's such a lack of images of this interaction. It's heavily understudied. So trying to find images to describe the system has been very difficult. So I took that into my own hands. Okay. I see some
Question
stuff.
about when our next meeting will be,
Oh,
but
okay.
not totally sure exactly which meeting that's about. The seminars are always the second Thursday. But yeah. And then we have the graduate student symposium and banquet coming up in April. So any other questions? Going once, going twice? There's a comment in the chat from Russell saying, thank you, really interesting research and wonderful presentation. Love the photos. So your macro photography is coming along great.
Yes,
Are you
thank
going to
you.
be moving on from extrafloral nectaries to other topics, other subjects?
You know what? I'm such a cactus lover. I don't think I'll move outside of that particular system. But there are so much more to uncover about extrafloral nectaries.
Oh
So
yeah. Oh,
if I
absolutely.
continue that research, I'll look into those other aspects like, nectar production through, different
Right. I was actually
values.
thinking in terms of your photography.
Oh, okay. Sorry. I got a question in the chat, threw me off a little bit.
Right.
Yeah, in terms of my macro photography, yeah. My next goal for this upcoming season is to get really good pictures of those herbivorous insects. Managed to find pictures of them on other cacti, but not on my own. And yeah, just get a couple more pictures of ants, the different
Oh,
species, because all of
cool.
the ones I featured in the talk today were the Chromatogaster depilus
Right. Okay.
species.
Yeah.
And so yeah, I want to get a couple more pictures with others. But
The recording, it's there's no kind of, Cecil here, no actual set schedule. It's usually, I would say, a week or two, but it's sort of just because, this is all done by volunteers, and it sort of depends on people's workloads and when they have the time to get to it. So there's a comment here. I hope that answers your question, George Z. There's a comment. There are lots of cactus and succulent societies that would probably be interested in seeing your talk. You could reach out to them. Thanks for the interesting take.
that's a very good tip. And I hate to say it, but I beat you to the punch. I gave this talk in San Diego to the San Diego Cactus and Succulent Society back in October, I think it was. It's been a couple months now, but yeah, I went down there and I spoke with them, and the San Bernardino, I think it's San Bernardino Cactus and Succulent Society also would like to hear from me, but I told them I would have to push it to next year. But thank you though, I really appreciate that.
I'll try to get the chat to you. It says here the Tucson Society pays speakers well.
Ooh, there we go. Yeah, I'd love to go to Arizona. I have family over there. So visit to Tucson wouldn't be too bad.
It then says Gates with an asterisk. I'm not totally sure what that means, to be honest. So I'll try to send you this, and you can see if you can follow up on it.
Okay, that sounds good.
All right. Well, with
Thank you.
that, any final questions? Oh, Gates for San Bernardino. Okay.
Oh, okay. Okay, that's what it
Yeah.
is.
But you will take a quick look at the I'll stay on, and you can take a quick look at all the love in the chat.
Okay, thank you. Thank you all. Thanks for having me. Much appreciated.
Thanks very much, everyone.
Oh yeah, I see now, Gates. Yeah, I think that's what it's called. I was given a business card for another one sitting right here.
Have you seen all the have you read through the chat?
Yeah, I saw some of the questions on here as well.
Okay. Yeah.
Yeah.
Well, you have to see, you want to get
Oh,
all the praise. I think I'll get sent a copy of the chat so I can send it to you. You can
Okay.
hang it on your wall or something. So any final questions from the people who are still here? Okay. Thanks,
Awesome.
Allie. That was great.
Thanks for having me. I appreciate
Great.
it.
Um,
All right. Bye, everyone.
okay. Bye-bye, everyone. Thank you.